नमस्कार दोस्तों, welcome to channel Toxicology on Board. Today in this video we'll be discussing another DABT topic, that is with respect to ICH S9 guideline. Uh, I couldn't find any previous ABT recertification questions on ICH S9 guideline, but as per current 2026 ABT brochure, ICH series of safety series of guidelines like S1 to S13 all are part of syllabus. So when I wrote the exam, questions were asked mostly on ICH S5 as well as S6. S9 is also very important guideline because current, if you see top five class of pharmacological class of drugs, in that apart from like antihypertensives or anti-lipidemic or weight lowering drugs, another important therapeutic area in the current market is cancer. So anti-cancer drugs are under large numbers which are being approved by the FDA or other regulatory bodies. So there are a lot of changes in with respect to ICH S9 guideline. Unlike the conventional molecules, small molecules like which were derived from the plant derivatives like Taxol or Taxane, new class of anti-cancer drugs are coming into market like tyrosine kinase inhibitor or JAK kinase inhibitor, ALK inhibitors. So, there is a drastic change in the case of guidelines also. So we'll be discussing this particular. So before any guideline comes into picture, this ICH regulatory body, what they do is that they give it in the form of question and answer also. First, they will publish a draft guideline. Then they will give you in the form. They will keep the guideline open for the pharmaceutical industries and various other industries researchers for them to come up with any question and answer. Next working document, they will try to answer this question and answer. Once all the question and answers are being addressed by the regulatory agency and series of discussion between the pharmaceutical scientists, industry scientists as well as the regulatory scientists, then the guideline will come into existence. I would also like you to go back and read about what is act, what is law, what is guideline. There is a difference is there. Guideline means it is not mandatory that you have to follow each and every step. Always another important thing you try to remember is whether you are in if you're a toxicologist and if you are in a position where you have to choose between guideline and study protocol, which you will be choosing, which you will give you more priority. So like that, these kind of questions you will be getting in an exam. So let's not deviate from the topic. So this ICH S9 main guideline I'll discuss in another video. Today we'll only discuss question and answer. So we'll discuss this particular question and answer document which was given in 2018. We'll discuss in terms of four parts. Okay, today this video will discuss part one. So directly we'll enter into the question. So first will be under section one, that is introduction. So there are series of 6 to 7 questions are there in this document which are related to section introduction. So first question is the ICH S9 guidance provides information for pharmaceuticals that are intended to treat cancer in patients with serious life-threatening malignancies. So whenever you are reading any ICH safety guideline, no, you have to see what is the scope of the guideline. It is applicable to what guideline? For example, here you see ICH S6 guideline is applicable to the pharmaceuticals which are biotechnology derived, when antibodies, antigens, this type of drugs. And if you look into ICH M3R2 guideline, the scope of this guideline is for pharmaceuticals uh, that is small molecular weight or small traditional type of molecules will be there. Small molecules के लिए this particular M3R2 guideline is applicable. So similarly, coming to ICH S9 guideline, it is applicable only to the anti-cancer drugs uh, for the treatment of the patients, cancer patients who are under serious life threatening, means almost they are about to die in another 2 months or 6 months. So to that type of patients, that type of patient treatment, those anti-cancer drugs, this particular guideline is applicable. All are initial developmental plans for anti-cancer pharmaceuticals covered under ICH S9 guidelines. So there was one particular person who raised this question. All the whatever the initial development plans are, they need to be followed only as per ICHS9. What the regulatory agency has given the answer, will see, as most initial development programs are performed in patients. Unlike if you look into M3 or other type of guidelines, no, healthy volunteers. If you go for phase one, you will go for. But here as the anti-cancer drug itself will cause cytotoxicity, itself its target is certain time DNA damage through which it is going to kill the cancer cells, or certain time these drugs cannot differentiate between the the host cells as well as the cancer cells, so they will target rapidly dividing cells. Okay, that is the reason most of the cancer patient you will see hair fall will be there, or melanin concentration, this because skin cells are also very rapidly dividing, or if there are any internal organs, their lining will be there, no, which are rapidly dividing cells will be there, no, these anti-cancer drugs will go and target these kind of rapidly dividing cells. So that is the reason we are not doing it in a healthy volunteers, rather this is applicable only for the patients who are having cancer. Okay, because the drug itself might have some safety issues. Okay, so because it might either cause DNA damage or cytotoxic in nature. So as most initial development programs are performed in patients, who are those patients? There are certain cancers which are only in the seen in the case of pediatric or adult, because cancer cannot differentiate. It can attack, it can be, there are a lot of cases where in pediatric also you can find this cancer whose disease is resistant, means there are no other treatments available, or even if there we have certain anti-cancer drugs available in the market, still we are going for the treatment of these cancer patients, still the patient is not responding, means disease is resistant, refractory to available therapy. Okay, non-clinical program described in ICH S9 is applicable. Okay, so for other initial development programs in cancer that is not resistant and refractory, ICH S9 should be used as a starting point, and other studies as appropriate with reference to ICH M3 guideline, that is M3R2, as well as ICH S6R1. Here we have seen, no, so I would request you pause the video, go back and read M3R2 guideline also, as well as ICH S6 guideline. Some questions you will be getting with respect to these guidelines also. In some situations where the development pathway is not clear, regulatory agency should be consulted. So always there will be pre-IND, IND meetings will be there, NDA meetings or like that will be there, type A, type B meetings will be there. You can approach them, you can ask them, they will definitely address you what kind of, whether I need to do 6 month study or 9 month study or 1 month toxicology study, do you need any additional cytotoxic test or immunotoxicity test. So like this you can write mails to these regulatory agencies and they will tell you, they will also suggest you. Okay, then next second question we will see, if the first in human study is conducted in a patient population with resistant and refractory disease. Okay, FIH study you're doing, that is phase 1 study. So you have to decide for this anti-cancer drug what will be the dose that can be given to this patient. Okay, but the patient is not respond. The patient population is refractory to that particular disease or resistant to that particular existing treatment. Will subsequent phase 1 studies in a different cancer but still resistant and refractory population still be covered under ICH-S9? Yes, because um, uh, will subsequent phase 1 studies in a different cancer, because here see there is another very good if you listen to this cancer doctors know, you will understand. Even if there are two people are there, if I have if for example if I have twins, I have twins. Okay, identical twins I am having, and first twin, for example I am having cancer, and my identical twin is having also cancer, but whatever the cancer is there with me, it is not identical to that of my identical twin. I mean to say, even for example in a vague manner, if I say there is a colon cancer, same colon cancer also in patient A is not similar to that of a patient B. Try to understand, that is the reason. So here what they are saying is that even it is if the it is conducted, if the FIH study is conducted in patient population who is resistant and refractory, subsequent phase 1 study different cancer still resistant, then still you have to follow ICH S9 guideline. I understand it is a bit difficult to understand, pause the video, try to read it. Okay, next question number 3, in general the guidance has been interpreted as applying when patient life expectancy is approximately 3 years. Okay, this is what the misconception is, that if the patient is having, if the doctor says you can for that particular cancer patient that you will not live beyond so and so 3 years, so it is an understanding that you have to apply ICH-S9 guideline to such kind of anti-cancer drug. It should be useful to provide further clarity about the intended population. So one of the from the one of the scientists is asking this question to the regulatory agencies. So please explain in clear, give more clarity on this internal population. So this was a statement given in the guideline, that is in section 1.3. Okay, go back, you keep the PDF document and try to keep it aside with you and try to read it. The ICH-S9 guidance does not make a reference to years of life expectancy. This much only, like 6 months only, 9 months only, 1 year only. They do not mention exact number. And the application of the guidance should not be based on expectation of the survival as measured in years. Okay, the intended scope of in the intent of scope is clarified in question number 1. Okay, it is applicable to what kind of life-threatening diseases for which the patient is not responding, refractory, those kind of anti-cancer drug ICH-9 is applicable. So whenever you are reading such these ICH series of safety guidelines, no, please read scope of guideline. This guideline is applicable to what? This guideline is not applicable to what? So one question with respect to this they will give you. For example, I will say you what they will ask you is that they will give you like this one case study they will give. There is certain scientist, okay, in a certain innovator company is there, and they are developing certain drug for so and so. For example, for and for weight lowering drug they are developing. So what are the following guidelines, what are the various toxicology studies need to be done, that toxicologists, so they will give you the option like 90-day studies, 1 month study, 6 month study, like that daily fertility study. So you should be aware of first how you try to address the question means you have to understand the nature of the drug. What is it, whether it is oligonucleotide or is it a aptamer, what is the origin of the drug, what is the chemical nature of the drug. Then you will decide whether it should be go for ICH S6 or ICH S11 or S12 S13, like that you will decide. Okay, so being aware of scope of each and every ICH S1 to S13 guideline is very very important. Next, can the principles of ICH S9 be applied to non-oncology therapeutics, means those drugs which are not used for cancer treatment but the disease is life threatening. For example, you have a HIV patient. We know if it is in the late stage of this thing, HIV has been detected, when there is the patient is not responding, life threatening is there, but that is not cancer with limited therapeutic options, and we do not have any options. For example, COVID also it was like that, it is a life threatening but it is not a cancer. So these indications, what is the answer given by the regulatory agency is the indications are outside of the scope of S9. Yes, because S9 very, very clearly it states this guideline is applicable only to the patients who are having cancer, and the cancer is also not responding to any existing treatment, and the cancer is refractory, it is relapsing, and life threatening. Patient survival is less than couple of months, 3 months, 6 months, 9 months, like that. Okay, see ICH M3R2 for guidance on when particular studies can be abbreviated, deferred, omitted or added. So if you read ICH M3R2 guideline, they will give you some kind of leverage or waiver they will give you. If you see what kind of studies can be omitted, where there are certain places you can avoid fertility study, segment 1 study, which you can incorporate in the repeat dose toxicology study only, or you can minimize this cancer study. Okay, so like that, case by case, on a case by case approach to optimize the drug development or life threatening or serious diseases than cancer. So this type of drugs also need to go for ISHM3. Again, if I tell you, for example, I was discussing, giving you an example of COVID also in this life threatening disease. COVID, if it is a vaccine, then you will not go for ISHM3. You have to follow CBER, that particular pathway. Okay, then with respect to that, you have to do the toxicology studies if they are needed. 6 month study or once in a 6 months administration, those type of study, and that too, that particular species should have a target which should be available in the human also. You have to relevant species studies, you have to do those toxicology studies also. Okay, so like that it is there. Next we'll see another question. So, are clinical trials in the adjuvant or neo-adjuvant setting covered under ICH S9? So, before that, you need to understand what is adjuvant, what is a non-adjuvant setting, okay, which will become a part, because most of the, as I told you in the beginning of the video, whatever the new anti-cancer drugs are coming now, the nature of them is not similar to the traditional origin. Traditionally means taxane, taxol, what was it, cyclophosphamide, carmustine. These were the drugs of option in the case of cancer treatment. But now you have tyrosine kinase inhibitors, you have what is one, ALK inhibitors you are having JAK kinase inhibitors. So there are new class of anti-cancer drugs are coming. Their origin is not similar to that of traditional molecule. More targeted therapy is there. They're attacking the targeted gene. Okay, some of them are aptamers, some of them are oligonucleotides. Okay, some of the antibodies are there, ADCs are there. So like that. So are clinical trials in the adjuvant or neo-adjuvant setting covered under ICH S9? This is the question which was asked by one of the pharmaceutical scientists. So what is the answer if given by the regulatory agency? We'll see. ICH S9 should be used as a starting point. Always in the previous, one more question also you have seen in the previous slide, they're asking use this as a starting endpoint. Okay, starting point for the drugs used in adjuvant or neo-adjuvant setting, even when there is a lack of detectable residual disease. Even after treatment, if there is still lack of residual disease is there, please use this ICH S9 as a starting point. Data generated in the patients when the initial program was in refractory late stages, patient was not responding, should be considered and may be used to abbreviated the non-clinical program. Then you can decide what are the studies can be omitted, or if the patient is still surviving. Initially we thought that patient will only survive 6 months, but now the patient is still continuing another 1 year also, then you will cut short, or what additional studies need to be done. In cases in which there is well understood high cure rate and low or long delayed disease reoccurrence state, then further studies, then you have to include cancer study. A complete program on reproductive and developmental studies are likely to be needed prior to the marketing. So for the initial phase 1, phase 2, phase 3, if it is not still needed, this long term cancer study or a battery of reproductive segment 1, 2 and 3 study, but once the drug is entering into the market, these type of studies also you need to be submitted to the regulatory agencies. In cases in which these factors are less defined and reoccurrence is high, the particular whatever the new adjuvant drug or adjuvant drug is not responding to the cancer a high or rapid, then the need for additional studies, their timing can be addressed on a case by cases, taking into account the totality of the preclinical and clinical safety data, cure rate and expected time to reoccurrence. Then you will decide whether, because here the main focus of ICH-S9 guideline is to not safety. Here it is time of the patient has to be survived. Even if my drug is causing some kind of severe adverse effects, whether if it is causing kidney damage, whether it is causing liver damage also, even then I'm okay with it, because I can put my patient onto dialysis, I can go for a liver transplant, or I can use some therapy which can be alternate the function of liver, whatever the function, whatever the main function of the liver is there. But surviving the patient, prolonging the lifetime of the patient is very very important in the case of ICH-S9 guideline. Unlike other ICH-S4 or M3 guideline, there safety is also important, efficacy is important, at the same time if safety is important. Here also safety is important, but primary is time, surviving the patient is important. Okay, weight of evidence approach you will be doing, risk benefit approach you will be doing. If the initial development program is in adjuvant or new adjuvant setting, additional non-clinical safety studies may be needed, including long-term general toxicology studies. Then you will decide whether I will go for 6 months or 9 months or 1 year repeat dose toxicology study, either in a rat or in the case of rabbit or in the case of a dog or monkey, like that. In a case, it is important to consider the natural course of the disease. What is the disease progression? You have to understand the pathology of the disease, how it is starting, whether we are able to relapsing is there, refractory is there. These things you have to see, nature and course of the disease. Application of this ICH S9, any omission of the study should be justified by the sponsor. If I am a sponsor, I am applying for this, for example, I am developing a new drug and the nature of that particular anti-cancer drug is neo-adjuvant or adjuvant, then by case, if I am omitting, for example, I am not doing fertility study, or if I am not doing any immunotoxicity study or some certain study, certain toxicology study, then I have to justify the sponsor why I have not done this particular study. If they are not doing fertility study, I'll justify to the sponsor stating that the effect of this particular drug on the male reproductive system and female reproductive system have been covered under repeat dose toxicology study. Like that, I have to justify to the sponsor. Okay. So stop here and read these particular sections also. Then we'll see question number 6. Question number 6, which was asked by the one of the scientists in the pharmaceutical industry, is in the case where a therapeutic increases survival, whatever the new drug which you are developing, if it is increasing the survival of the patient. Initially, the patient was taught that he will only survive 3 months or 6 months or 9 months, but now he is surviving 1 year, 1.5 years. What further toxicology work is recommended? What is the appropriate timing of any studies? Again, if before going to the answer, what the regulatory agency has given the answer, my answer would be case by case only. You have to see, because as I told you, no same cancer is also not same in two different individual cancer patients. So let's see what is the answer given by the regulatory agency. When the anti-cancer pharmaceutical is shown to extend the survival of the patient, no additional general toxicology studies are usually warranted, because already you are testing the drug in the patients, humans. So establishing the safety in animal at this point is not needed, but we'll see what are the exceptions. The clinical safety data in the intended population is more relevant to assess human risk than those generated in the animal studies. As I told you, already you are given to the human beings, you have ample amount of data is there, safety established in the case of human. So at this point, animal additional general toxicology studies in animals is not needed. That is what the agency is saying. Then additional toxicology studies other than general toxicology may be needed on a case by case. For example, I think they're asking for immunotoxicity or some kind of things. We'll see what is there. If additional studies are deemed important, such studies could be submitted post approval of the anti-cancer pharmaceutical. So stop the video and try to understand what is this one. Whatever I could say, I have explained, I have discussed with you. Now we'll enter into the question number 7. Okay, under introduction section only. The scope indicates, as I told you, always the safety series of guidelines read scope. Okay, the scope indicates that in patients with long expected survival, similar to that of the previous question, the recommended for additional non-toxicological general toxicology studies depend on the available non-clinical and clinical data and the nature of the toxicities observed. So in the patient, if the patient is showing some kind of adverse effects, you have to make observations of these. Then our additional non-clinical safety tests are needed when an anti-cancer pharmaceutical in the clinical development or approved for a particular malignant tumor according to ICH guideline. For example, it was accepted for liver tumor, is to be applied to another oncology indicate. Now this same drug investigational, whatever the anti-cancer drug, I am using it for pancreatic cancer. Okay, that is not immediately dangerous. For example, this earlier liver tumor was a malignant tumor, more dangerous. Now the same drug I am using it for pancreatic cancer, and that particular cancer is not serious. Then how now this is not coming under ICH guidelines. So what additional studies? What is the agency is giving the answer? As per my understanding, new studies you have to do. But let's see what is the. I might not be right also. So when moving therapeutic development from an approved indication in oncology or from an approved indication with an sufficient non-clinical and clinical safety data set to an unapproved oncology indication. Here in this case, pancreatic cancer, which is not approved. That is not immediately life-threatening, but it is serious. Additional general toxicology studies, example chronic studies, 6 months or 9 month studies are generally not warranted. So it is saying it is not needed. But my understanding was probably it might be needed. But no, this is not needed. Why? We'll see what is the explanation there. Similar to the response under section 6. So go back and previous slide you see. The clinical safety data. Yes. So the key point here it is that since already the drug has been tested in the patient, so additional general toxicology studies are not needed even if you are changing the indication. See bottom line again, initially the drug, there is an investigational drug. It was developed for liver tumors, okay, malignant tumor, okay. At the time, uh, entire battery of general toxicology studies was not done because it was life threatening. Now you're taking the same investigational drug, you're using it for second type of tumor, that is pancreatic tumor, which is less serious, which is less life threatening. Now the question is whether we need to do all type of general battery of toxicology tests. The answer is no, not needed, because already the drug has been tested in the patient. So now doing additional animal studies is not going to add any kind of additional information. So see, main thing if you look into 3R principle, that is the reduce, refine, recycle. No, what it is saying, if you're having a data, use that data, whether it is clinical data, epidemiological data, any data. If by doing additional studies, if it is not going to add anything new to the existing information, then avoid it. type of studies, whether it is animal studies or whether it is in vitro study or any kind of studies, because reduce 3R principle you need to clearly apply to this type of guidelines. So we'll see last two statements similar to the response under question number 6. The clinical safety data generated in the patient population for the approved indication, for example it was liver tumor, are most meaningful and relevant to inform the safety plan for the patient population in the approved indication, that is pancreatic cancer. Toxicology studies other than general toxicology may be read on the case by case. So here it is not clearly mentioning what additional toxicology. I am assuming that it might be local toxicity or some immunotoxicity, such kind of things might be there. Okay. So hope from this video you were able to learn getting introduced to the ICH S9 guideline. Question and answers we have discussed. We have only discussed section 1 under introduction. Only those 6 to 7 questions we have discussed. In another video, we'll discuss part 2. Other sections 3, 4 also we will be discussing. Okay, thank you so much. Be curious. Read ICH S1 say S13, all the guidelines, scope you read, applicable to what, not applicable to what kind of studies need to be done, what kind of studies need to be submitted to the regulatory agencies. Try to read it, try to remember it. That only questions they'll ask. At my time, they asked me about S5 and S6. Probably some other students they might be asked about immunotoxicity, S8 also like that. Okay. All the best for your exam.